The second point is that our system is using a bidirectional flow of solution. So I will introduce more later. So it will, with this technology, it will increase the extraction efficiency and the recovery rate of the nucleic acid. This system is very easy to use. It has a touch screen panel right over here. So, and it will be used with some prefilled cartridge, so you don't have to set up anything. You just simply snap in a prefilled cartridge and the samples, and then everything will be ready. The system, you do not require to batch any sample. You can start to run from 1 sample up to 24 with our system. You don't have to batch it. You can use it for 6 samples, 7 samples, 8 samples. So and other selling features of this product, and it has a very wide specification. You can use it with different multiple DNA extraction kits. So let's start with the technology first. Is everyone see the screen of my PowerPoint? Okay, so is it okay for everyone to see my PowerPoint? Oh, yeah. Okay, so let's start with the second page. So let me introduce more specification about the characteristics of the column-based extraction. So in the market right now, you will find some small extraction system. For example, Quadrant or MacPurix. Other extractions, most of them are using magnetic beads. But what is the difference between our system and the other system? So with magnetic beads showing on the left-hand side, magnetic beads will involve a lot of agitation. So you require the magnetic beads to shake the samples and to lyse the samples. So in this process, the agitation may destroy the nucleic acid integrity, so by the shearing force. However, our column-based system will have because it's utilizing a membrane, silica membrane, so it will remain the DNA integrity. For the magnetic beads, also, I will demonstrate later. The concept of magnetic beads is something like a fish hook. So one hook for several fish or one fish. But however, if you're using our membrane-based extraction, it is like a fish net. It's a net to capture all available nucleic acids. And the third part and the most disadvantage of using magnetic bead is that it will have the carryover problem, which I will demonstrate later. But with the column base, because it's simply a column membrane, and all the purity will be washed away and be collected in the collection tube. And then all the purity will not be carried over. So there are lots of benefits using column-based extraction system. Let's go for it one by one. So, wait a minute. Okay. So actually here it should be some animation, but however, right now I have uploaded the PowerPoint to the server, so the animation cannot be shown. Right now I will just demonstrate with my pen tool here, so to give you a more better picture about the differences between magnetic beads and Puro membrane column system. So here you should see that here actually is a, you can imagine it as a sample. So here you have some impurity like this one. This is the impurity. You have the DNA here. And also you have the magnetic beads here. So during the extraction process, the magnetic beads will agitate, it will shake in order to destroy the cell and release the DNA like this one, in this direction, like it shake. However, the DNA here will be disrupted or damaged by this shearing process. So the nucleic acid integrity will be destroyed. If you are using a membrane-based technology like the Puro system, all the impurity will be washed away. Okay, will be washed away will be passing through the membrane directly, and the nucleic acid will remain intact on the surface of the membrane. Thus, the integrity of the nucleic acid will be maintained. So this is one of the benefits of using the membrane-based nucleic acid extraction technology. So another point is that the fish hook and fish net concept. So if you're using a magnetic beads, so for example, there are 2 beads here. Of course, in the whole extraction sample, you have a numerous of magnetic beads. But nevertheless, one magnetic bead can be able to, for example, bind several DNAs and then it will leave. So like here, maybe it will bind 1 or 2 DNAs out or several different DNA. However, you can imagine that it is like a fish hook. It can only fish out some DNAs out, but there will always be remaining some DNA inside the sample, which means that the recovery rate of DNA will decrease in this concept. However, with the column-based system, the membrane acts like a fish net. So all the DNA will remain here, will remain on the membrane surface, and the impurity will be washed away. So the membrane serves like a fish net to capture everything, to capture all the DNA in the sample. So it will have a higher recovery rate. So another features of our product using the Puro system is that inside the machine, when you're comparing with some traditional column-based extraction, the DNA only pass through the membrane, okay? Pass through the membrane in one direction. Okay, the DNA will pass through the membrane here and then in this only one direction. However, in our system, we are utilizing a different kind of solution flow called the bidirectional flow. The solution will pass through in this direction and the machine will pump back the solution up several times. So you can imagine that if there is any escaping DNA passing through the membrane, it will bind back at the other side of the membrane. So we are actually utilizing both surface of the membrane to increase the extraction yield of the membrane-based column. So we'll have a high extraction yield. Here shows you some data. So we are actually comparing our Puro system with an M-brand magnetic bead extraction system in the market. So in this study, we are trying to extract some cell-free DNA from some clinical samples. So you can see that we have a significant higher cell-free DNA extracted in the samples. Look at the right side here, we are also detecting the GAPDH, some housekeeping genes. You can see that the CT value detected with our system is lower than the M-brand, which means that we have a higher copies of housekeeping gene detected, which shows that we have a much better extraction efficiency when you're comparing with some magnetic beads extraction system on the market. So the second point is that it's about the carryover problem. And let's imagine here. Here is the magnetic beads. It will go through here. It will extract some DNA and then pass to here, and then it may wash. You will have some magnetic beads here because it's binding DNA and the DNA here. Okay, it's some DNA here, and then it will pass another. After washing, it pass another to another well and then extract out the DNA. During this process, you can imagine because the binding efficiency between the magnet and the magnetic bead is not it can never be 100%. So some magnetic beads may be carried over to the samples itself. So you will always find that no matter which system you're using, in the samples, you will also find a trace amount of impurities in the magnetic beads being carried over to the samples. So it will cause a serious problem of P-cell inhibition or P-cell inhibitors. However, with the Puro membrane, as you can imagine, it is always sort of like a fishnet, and all the impurity will pass through, all pass through the membrane, and then these impurities will be removed by physical separation. So, and what you get is a very pure DNA extracted. So you can forget about the carryover. We do not have such kind of problem. So regarding this, we also have some data to support this argument. For example, here you shows you the differences between the sequencing result with a magnetic bead extraction system and the Puro system. So you can see that the sequencing data, the peak shape is much better than the magnetic bead system. For example, you show it here. Even for A signals, at the right-hand side, you can see there are some missing because due to the PCR inhibitors. Here, you shows you a very nice peak of A. However, due to PCR inhibition, you can see the very worst graph of sequencing signals here. Okay, so this actually shows you the quality of the DNA extracted with our membrane-based automatic Puro extraction system when comparing to the magnetic bead systems. So, we also performed a dilution test with comparing our system with the Q-brand system. It's also a magnetic bead system. So here will require a little bit of explanation. So we have diluted the sample tenfold for 2 times. So here is the undiluted samples. Here is the diluted sample. So you can see that from our system, the CT value between undiluted sample to a tenfold diluted or 100-fold diluted samples are similar, basically the same distance, which means that it is equally diluted. However, when you're looking at the CT value of a Q-brand system, you can see that the undiluted samples is right here, okay, at this point. Here, they diluted. We got the same CT value with the tenfold diluted system as the Puro extracted DNA. 100 with 100 dilution is also the same with the Puro system. And this graph, actually, you can imagine the concept in this way. For the Q-brand system, because it has the inhibitors inside the samples. So during the undiluted sample, you will have a lower CT value, okay, higher CT value, which means that there is some sort of PCR inhibition, so delaying the amplification cycle. With a tenfold dilution, the PCR inhibitor is diluted, so it will decrease the PCR inhibition, and then achieving a similar CT value with the Puro system, and so forth, like the 100 diluted is the concept. So this graph actually shows you that the Puro system here, we have a higher PCR inhibition. It's not due to the sample quantity we input for the PCR, but that is due to the inhibition, because when we diluted it, we achieved the same CT value as the magnetic bead extraction system. So the differences between these 2 points, the undiluted sample by Puro system and the undiluted sample by the Q-brand system, the differences is due to PCR inhibition, but not the sample quantity we input at the beginning of the PCR process. Yet this is another example showing that the DNA purity that extracted by the Puro system is much better than the magnetic beads extraction system. So we also have some more data telling you about the purity and the yield of the extracted RNA. Our system and our kits are also able to extract RNA. So please take a look at here, which sees that during the when we are looking at the RNA purity, we are looking at the 260 to 280 OD ratios and 260 to 230 ratios. Both of the ratios achieve around 2, which means that for the RNA is a very good range. The purity of the RNA is very good. And we also subject these samples to some quality checking method. So the RQN, the RNA quality number achieved from the samples around 10, which is a very, very good integrity number. So RQN number, we calculated from the 28S to 18S ratios. So we found that the RQN achieved by our extracted RNA is very good. This is around 10. The maximum number is 10. So the extremely high quality of RNA extracted can be shown with our extraction system. So also the reproducibility of our system is very good. This graph showing you 4 different samples. Okay, this is 4 different samples. You can find that the variation here is very small, and the CV is less than CV. The variation percentage is less than 5, which is a very good reproducibility, and the reproducibility between manual system and our automated system is the manual system. Automated system is also comparable. So you can really purchase our automated system and the manual system. They both perform equally good. So from here, is everyone clear about the technology that we are using right now? If everyone is okay, I will go on to introduce more features of our systems right here. Is everyone clear about the technology so far? So right now I'm waiting for some feedback. Okay, so I see Matthias say yes. Okay, so I will go to introduce more about the system design. So here shows you the cartridge of our system. It is a prefilled cartridge. The reagent well here is the reagent well, and here is the heat well. We will aliquot some during the whole process. We will aliquot some reagent here and do the heat incubation right here. So we have column tubes. Our columns is right here. The membrane column is here, and we also have some filter adapter. So you can see that the whole workflow from lysis to binding to the membrane, washing, drying to elute is performing on the same cartridge, which means that you do not require to set up any reagent bottle or to set up any to do any pipetting. You just simply snap in one of the cassettes for one sample. This is very convenient to perform. So in other feature is that the inside of the system is like this. So you snap in the cassette here. They are operating in separate channels, which means that you do not have to batch the sample. You can, if you have like 2 samples on hand, you can work with it. You just simply snap in 2 prefilled cartridge and put in the sample right here, and then the experiment can be done. You don't have to batch anything. You can perform from 1, 2, 5, 6 to 12 with our Puro 12 machine, or if you're buying a higher throughput system like a Puro 24, you can run from 1 sample to 24 samples. So you don't have to batch. And other benefits of individual channel is that you do not have to worry about a systematic contamination. So everything is working individually, so the system contamination will be reduced. So the interface is something similar to this. It is very easy to use. It will guide you through everything. And then here only you only have to require set up sample numbers 1 to 12. Elution volume, you can choose from 50, 100, 150 to 200 microliters of elution volume. You can select different kits here, and then you can start the run. It's very easy to set up the procedures. So talking about the compatibility of our extraction kits. So right now we are going to launch 3 different extraction cartridge, which will be used on our Puro system. The first one is called the blood DNA. It is specialized in extracting blood samples. You can do it with whole blood, body fluids, culture, plasma, serum, and buffy coats. The second one will be tissue DNA. It is what we are calling it a universal extraction kit. So you can use different types of sample types with extraction cartridge. For example, urine, swab, LBC, cytology, bush, saliva, dry blood spot, nails, hair, feathers, bacterial cultures, bacterial in body fluids, positive cultures, avian insects tissue or animal tissue. A lot of different kits can be. As for stool sample, we haven't validated it yet, so I have to check it back. But so far, right now, we don't have the data for the stool samples. I remember that we may have some alternative extraction kits specialized in the stool sample, but I have to check it back. So the other is the third one will be the Pure viral DNA RNA extraction kit. It is a co-extraction kit, which means that for viral DNA, if you are extracting viral DNA or viral RNA can be extracted all together. It's a co-extraction kit. So it works with plasma, serum, cell-free body fluid, cell culture supernatant, and animal tissues. So large variety of sample types you can input to this system. So you don't have to worry about buying different types of sample. Of course, we are going to launch different kits later, which are specialized in some special sample types, for example, FFPE with a better extraction quality. Okay. So talking about the compatibility with GenoFlow series. So for blood extraction kit, we've tested it can be done with our thrombophilia test kit or beta thalassemia array test kit. For the tissue DNA, you can work with the HPV and STD array test kit. For the viral RNA DNA extraction cartridge, you can work with the HCV and HPV array test kit. So it is highly compatible with our own GenoFlow DNA array series. So this extraction system is an open platform, so which is a little bit different from S-brand competitors in the market right now. Our extracted DNA can work with different types of diagnostics platform. For example, you can use it with Roche, or you can use it with Fluidem. Of course, you can use it with our FTPO system and also CSF system. So it is an excellent extraction platform that you can put to all of the labs. So if this is okay for everyone, right now I would like to go to a little bit of comparison. I have chosen two brands for the comparison. One is Biomirix, which we are comparing with EasyMac, and the second one is QuietTube. So let's start with EasyMac from Biomirix first. So here I showed you some pre-treatment step. The setup step of EasyMac between EasyMac and GenoPure extraction. So here, if you're using EasyMac, first you have to batch the sample. So because the cartridge they're using are for X samples, then you have to do sample pre-treatment because it is an open platform, and then you have to do sample login, reagent loading to the machine, and then you load the cassette to the machine, and then you have to do onboard lysis of the samples. And then you have to add the silica magnetic beads by yourself, and then the machine will do extraction automatically. However, if you're considering the manual manipulation during the whole process, our Puro system are much less. So you can see that with our system, you only have to do sample pre-treatment, and then you log in the sample, and then you load the cassette, put in the sample, and then the extraction, you can start the extraction right away. So we have much less manual manipulation during the system, during the extraction. Here shows the nucleic, the EasyMac extraction cartridge. So each cartridge have 8 wells, so you have to batch at least 8 samples for 1-1. But however, we are using individual pre-filled cartridge, so you do not have to do batching. So the pre-treatment, you cannot find it is not very well supported for the pre-treatment process. So but however, in our manuals of our extraction kit, we have stated all the reagents and protocols required for sample pre-treatment. For example, for urine, if you're doing the STD test, we will suggest a series of easy sample pre-treatment before you load the samples to achieve a better extraction result. So here, another point of reagent load is the reagent loading. In our system, we do not require that because all the reagents are pre-filled in the cartridge. But if you're using EasyMac, you have to load the reagent yourself to the machine, something like here. So the machine is using the reagent filled in these large bottles for each extraction, which actually will have raised a problem of system contamination. So what if the bottle is contaminated? So everything that you extracted will also be contaminated. And of course, you require to set up a lot of different things. But in our system, you only use a pre-filled cartridge. So the manual setup is not needed to set up any reagent bottles. And we also reduce batch contamination because all the reagents are separated from sample to samples. Okay, so and other things that EasyMac has to be done is what we call on-board lysis, and you have to add the silica magnetic beads yourself. So here, you put in the samples, you put the cartridge to the EasyMac. You have to wait a little bit and for it to lyse the samples here on board is what we call the on-board lysis right here. After the lysis, you have to manually pipette the silica beads to the system. So this is a very manual way to do it. However, in our system, you just simply snap in the cassette, put in the samples, and then you can walk away. You don't have to do all the incubation and lysis and the column loading to the platform is all automated, so you can just walk away. So here we shows you that the Puro system is much automated than the EasyMac system. So and actually the reagent and machine costs are relatively high. We found the information here from a web. Some of them are least price. You may find a cheaper one in the market right now, but however, the cost you're calculating is around 6.7 USD per reactions. So I cannot disclose our selling price right here because this video is going on public, but you can contact our sales representative, and then you will find we actually are much cheaper when you're comparing with EasyMac. So the second comparison we are doing will be the QuietCube. So QuietCube from QuietGen, as you all know, they are also using column-based extraction. So what is the difference between these two systems? First thing, for QuietCube, you have to batch. If you have the experience of using QuietGen, it is very complicated. I'll show you here. For example, when you have two systems, you have to put on the rotor like this. So in QuietGen, they are using rotor instead of vacuum pump or air pressure to conduct the solution flow. So they are using a rotor. So basically, QuietGen is something like a manual step. You put manual extraction, but they put the rotor inside the machine, and they have a robotic arm to move the solution and the column. So it is like a manual extraction step, but everything put inside a machine. So when you have the experience of using QuietCube, you can see that for example, there are two samples. There's two samples. You have to set it like here. 3 samples you have to set it like here to maintain the balance of the rotor. 4 samples, 5 samples like this, 6 samples. You cannot run with 11 samples. You cannot run with 1 samples. So there are a lot of very complicated. The setup and the batching system of QuietCube is very, very complicated. You have to also set up the reagent in this diagram. You cannot set otherwise, or the robotic arm cannot pick up the reagent. So here is the reagent tank. You have to set it up like this. Okay, this is the sample, so you have to set up like this, exactly according to this table. So this is a very complicated way to set up the system. However, as I have explained before, our system, you can put it in like 1 sample, 2 sample, 3 sample. You do not have to batch anything. And you do not have to worry about how to put your reagent on the cartridge. So because everything is working in individual channels, so you just simply snap in the pre-filled cartridge, and then everything will be done automatically. So the second point is the loading reagent and disposable. Here shows you the inside of the QuietCube system. Here, the rotor I just mentioned, sample, reagents, and the pipette tip. So here they have a robotic arm which move in 3 axes. So in 3 axes, this 3 axes, okay. With more access, more complicated a robotic arm move, it will increase the chance of a contamination because how it moves is actually it takes the pipette tips here and then extract here and then put in here. So there are lots of different movement inside the machine, which will cause contamination, have the risk of causing contamination. But in our system, we only have two axes, like back and forth, up and down. So we reduce one axis, which reduce the contamination risk. So as you can see, QuietCube actually is a very complicated machine to set up, and it requires a lot of batching and how to put in the sample, put in the reagent is quite difficult. But with our system, we are much more simple. So everything is pre-filled in the cartridge, and you can load it with one sample, two samples. You do not have to batch anything. Both